Let's focus. How to study cells. Imagine trying to read a message written with dots so tiny that your eyes can no longer tell whether there are two dots or just one. Our eyes are powerful, but they have a limit. When two objects become extremely close, the human eye cannot separate them clearly. This is called the limit of resolution of the human eye. The limit of resolution is the minimum distance between two closely spaced objects at which they can still be seen as separate and distinct. The limit of resolution is the minimum distance between two closely spaced objects at which they can still be seen as separate and distinct. When you hold something about 25 centimeters from your eyes can see two tiny points separately only if there is a small gap between them. If the gap is less than 0.1 millimeter, your eyes cannot tell them apart. So they look like one single point. The limit of resolution of the human eye is 0.1 millimeter. Now think about a cell. A cell is far smaller than 0.1 millimeter. In fact, most cells are completely invisible to the unaided eye. So, this raised a fascinating question in the minds of the earlier scientists. How to study cells? In our minds, the query that arises, if cells are too tiny to see, how did scientists discover them? Yes, they used the microscope. The trees, the hen eggs, etc. can be viewed by the naked eye. But the electron microscope helps us to view atoms, lipids, proteins, etc. While the light microscope, that is less powerful than electron microscope, helps us to view bacteria, virus, etc. Using convex lenses, scientists learned how to magnify tiny objects and reveal an entirely hidden world. A microscope uses combinations of lenses called the objective lens and the eyepiece to enlarge objects thousands of times. Let's travel back to the year 1665. A curious scientist named Robert Hooke examined a thin slice of cork using a microscope that he designed himself. What he saw amazed him. Tiny box-like compartments arranged together like little rooms. He called these structures cells. That single observation became the foundation of modern cell biology. Today, in school laboratories, we use light microscopes with different objective lenses such as 10 times and 40 times magnification to observe tiny structure under visible light. Through the microscope, ordinary objects suddenly reveal extraordinary details. Some schools have electron microscope too. So, why is electron microscope powerful? These instruments use a beam of electrons instead of light to produce highly magnified images, allowing us to see cell structure at the nanometer scale with remarkable clarity. A nanometer is 1 billionth of a meter. But here is something interesting to think about. How to estimate the size of a cell? It is a lab activity. Imagine trying to measure something too small for your eyes to see. We will consider an onion peel cell. Since you can't hold a ruler up to an onion cell, you have to measure the background field first and use a little maths. Here is how you calculate a cell's real size in 3 simple steps. First, measure the field of view. Place a transparent ruler on the stage of the microscope. You can focus on it using the adjustment knob. Look through the eyepiece to see the bright circular field of view. Measure the width of the circle using the millimeter marking. If it measures 5 millimeter, convert it to micrometers, that is 5 into 1000 equal to 5000 micrometers. Second, count the cells. Swap the ruler for your onion peel slide. Look at the cells lined up in a straight line across the exact center of that circular field. Count how many cells it takes to get from one edge to the other. Let's say you count exactly 25 cells. Now the maths. That is the third step. To find the width of just one cell, divide the total width of the circle by the number of cells that fit across it. That is, estimated size of the onion peel cell is equal to diameter of the visible field in micrometer divided by number of cells along the diameter. By using a standard ruler to measure your field of view, first you can easily figure out that a single onion cell is 200 meters wide. The total magnification of a microscope depends on two lenses, the eyepiece lens and the objective lens. The formula of total magnification is equal to eyepiece magnification into objective magnification. If both the eyepiece and the objective lens have a magnifying power of 10 times, then 10 times into 10 times is equal to 100 times. So the total magnification of the microscope is 100 times. This means the onion peel cell appears 100 times larger than its actual size when viewed through the microscope. The lab activity is not only interesting but informative. A microscope helps us see extremely small structures clearly and play an important role in studying cells and their structure. Over time, scientists improved microscopes by enhancing three major features. The first is resolution, which means clarity. The second is contrast, which is the difference in brightness between different parts of an object. The third is magnification, which makes tiny objects appear much larger. Remember, the three main features of microscopes are resolution, contrast, and magnification. Resolution, contrast, and magnification. If these features are improved, microscopes becomes powerful. Here you can see an image of the lower surface of a Colocasia leaf observed under a scanning electron microscope. This advanced microscope produces highly detailed images of surfaces revealing structures that cannot be seen with an ordinary light microscope. We understand the technology, especially the microscope, has helped us learn about cells. Like, share and subscribe for more NCERT science explanation.